Tanya Siddiqui presented some data with CLL. I thought that was pretty encouraging. This was from the phase 1 study with lisocabtagene maraleucel over the dose escalation part of the study, and showed again a reasonably high complete remission rate, and encouragingly development of MRD negative remissions by even deep sequencing, and that many of those remissions continued to improve even out past 6 months. And it seems like a lot of those patients that were only, quote, PRs, we had very bulky disease, and it's very hard to get less than 1.5 centimeters to qualify as truly a CR by IWCLL criteria. So we'll have to see what happens, but I think that was very encouraging. I know the phase 2 study in CLL is ongoing with that agent.
What I think is also very, very encouraging to see that you also can tweak the immune system by actually pre-treating these patients with ibrutinib. That was shown at ASCO quite nicely. So it just gives off a pathway that we can actually improve on actually our immune system, the out product that we generate by actually pre-treating the patients with immunomodulatory drugs.
Yeah, I think they're hoping to present that cohort at one of the next meetings as well. Okay, so we're talking about heme malignancies. What about solid cancers? Anything on the horizon?
Yeah, here we are at the American Society of Hematology annual meeting, but the reality is cellular therapies are often administered in transplant programs and large academic centers that are familiar with cell therapies for blood cancers. And we're now seeing that we're helping to facilitate the treatment of patients with solid tumors. And I think that CAR T-cell therapy for solid tumors is a little more difficult than targeting CD19 or BCMA. The expression of molecules on the surface of most solid tumors, those molecules are often expressed on normal tissues, so the toxicities our rates may be higher, and there's also issues of trafficking into the tumor. So I'm actually more encouraged by data from tumor-infiltrating lymphocyte, or TILs therapy. There was data presented at the ASCO annual meeting this year showing that melanoma patients who were refractory to checkpoint inhibitors were able to have tumors removed and tumor-infiltrating lymphocytes grown out and actually have responses and durable responses to those therapies. And similarly, patients with cervical cancer were having responses. And so we're very encouraged, and at our center we help facilitate those treatments, and I believe that we may see FDA approvals for TILs, and that may become a platform to gene-modified T cells as well in solid tumors.
I think you're pointing out, though, that the tumor milieu is not the same in a solid cancer than it is in a heme malignancy, where we're seeing these dramatic expansions and likely cures. And so we've got a ways to go, probably with T-cell engineering and the like, to try to figure out molecules.
TILs have been impressive, but they've been impressive for some time, and we have to figure out why these cells are infiltrating the tumor. In melanoma, we've seen that, but it's curing or having long-term responses in a minority of patients. Figuring out what is it about the tumor, or what about the T cells causing them to infiltrate, and then, as you said, making those into gene-edited cells so that you're actually producing those at scale, as opposed to having to go through the whole exercise of producing buckets worth of tumor-infiltrating lymphocytes.
Okay, let's get out the crystal balls here, and everybody can look a little bit about at the future. So how do we? These are early generation CARs, and so I think we're going to see rapid changes. What are the kinds of things we're going to see going forward? Do you predict?
I think the idea of the so-called off-the-shelf CAR T cells is very attractive. One of the challenges with CAR T cells right now is having to wait, and we talked a lot about bridging therapy today. If you could treat someone with a CAR T cell later this week, or even the next day after you plan to do that, much in the way you can do with chemotherapy, that would have a lot of advantages. There are other advantages of off-the-shelf, but that time frame shortening would be very attractive.
Yeah, so some of the other advantages, we also talked about T-cell fitness, and so if you take a healthy donor who doesn't have a cancer, which interacts with the immune system and also hasn't seen chemotherapy, that is potentially very important. But on the flip side of that, giving someone else's T-cells increases the risk that those T-cells will be rejected potentially before they can have an anti-tumor effect. The other advantage to off-the-shelf CARs is that from one donor you can make up to 100 products, right? And so that is that's intriguing when we're talking about the cost of this therapy.
But you have to then deal with the risk of causing graft versus host disease.
So most of these off-the-shelf CARs have been engineered in a way to try to limit the risk of graft versus host disease. So they all involve gene editing technology that edits out the TCR and edits in the CAR in its place. And so I think we have seen, I think, enough data to this point, although it's all very immature and it's all very small studies, where the risk of graft versus host disease, thankfully, is incredibly low and really limited to sort of transient rash, which, to be honest, may also just be a sign of CRS, right? So it's hard to know.
What about other products other than T cells?
Well, I think NK cells are a very attractive approach, and there's some emerging data using NK cells as a platform to being engineered to deliver cellular therapy. And I think this is really interesting just to look at the toxicity profile that they might induce. It might be that the early data shows that the neurotoxicity is definitely less using NK-driven products, but obviously the trial data has to evolve.
Jason, I think you guys have that data from
The allo-NK, yes, and with the IL-15 as well built into it, and it seems very promising so far with low toxicity rates. Obviously, we need more patients and more data, but yes, it is intriguing.
I think one of the most impressive things on that data set is that there were several Richters transformation from CLL that were treated, and there were at least 2 CRs in Richters, which is not something that we've seen.
That was data from Katie Rosvani's group at MD Anderson with the cord blood-derived NK cells with co-expressing IL-15, which was a very interesting trick. So that's really good. Okay, we still have to deal with relapse, and so how are we going to do that?
Yeah, so one of the things that I'm most excited about is starting to understand, or looking to understand, the biology of these relapses. We understand there can be loss of target antigen, and CD19 treated ALL, maybe up to 50% of patients who are relapsing could lose CD19. In large B-cell lymphoma, about 1/3 of patients who are relapsing are losing CD19. So dual targeting CARs may help to increase the durable response rates, but I think we're starting to see emerging data, both from our group and also from some of the pivotal trials, that there are suppressive myeloid cells that may contribute to lack of durable responses in these patients. So strategies that could help mitigate those suppressive myeloid cells may also be important.
What about combinations? The obvious is checkpoints, right?
Yes. So the ZUMA-6 trial combined axicabtagene ciloleucel with atezolizumab, which is a PD-L1 antibody, and we presented the results from the phase 1 portion last year at ASH and await the results of the expansion in phase 2. But in the phase 1 portion, 12 patients were treated. The response rates, it's really hard to compare 12 patients against 100 patients on ZUMA-1, but the response rates were at least as good, if not maybe the slightest hint that they were better. What was interesting on that in that cohort is that there were at least 3 patients who had conversion from a PR or stable disease out past 6 months. So there was a delayed conversion of PR and stable disease to a complete response, which is hypothesis generating. I think that maybe these cells are sticking around longer and being revitalized and being able to treat active tumor. And that was borne out by looking at just the area under the curve of T cell expansion, and the T cells did seem to persist at a higher rate for a longer period of time. So
I think we're looking forward to the readout of those trials. Yeah. But it also sets the stage to add ibrutinib, lenalidomide, maybe even bispecific antibodies based on the study we saw at the plenary session, where patients basically receiving a CD20, CD3 antibody had even activity after failing CAR T cells. So thought-provoking.
Thought-provoking. Obviously small numbers so far, but yes, there's a response rate there where we don't yet have any therapies that are looking that promising, so we need more data.
All right, thank you very much for your attention. This has been very informative, and I thank you. On behalf of our panel, we hope that you found this peer exchange discussion to be useful and informative for your practice.